Hello students. So today we are going to learn a new topic, that is plant growth and mineral nutrition. Chapter number 7, part 1. In this part of the chapter, we are going to learn plant growth, phases of growth, conditions for growth, growth rate and types of growth, also growth curve, differentiation, de-differentiation and redifferentiation. So based on the topic, I kept a simple animation image in the first page. Now moving further, second page, we can see a simple seedling or seed germinating, right? And second half of the slide, we can see the plant with fruit, the fruits with seeds, further planting the seed, then germination, sprouting, seedling and plant, plant with flowers, and finally again plant with fruit. So simple cyclic formation of the growth of the plant, right from seed to the plant with the fruit. Now third slide, again I kept a simple image to make you understand the concept, that is seed germination. So seeds, when they germinate, initially the formation of radicle takes place, further the hypocotyl and the root moving towards the soil, whereas above the soil the formation of cotyledon and the seed coat getting withered away, further the seedling formation with hypocotyl, epicotyl and the cotyledons, and also we can see the first true leaves. Then coming to the introduction part of the chapter, plant growth. Now here I added a simple animation image. Now you can see the children sowing the seed and becoming so happy in seeing the growth of the plant. Now even that is an animation where the plant, right from the seed to the seedling to the growth of the plant, taking place in quick succession, but rather it takes place a longer time. But seeing this image, we can understand the happiness seen in the children's face when they see the growth of the plant. Now first point, growth is one of the characteristic features of living organisms. Growth as a phenomenon has two aspects, quantitative, qualitative. So as we know, growth is an important aspect for every living organism, whether it's a plant, whether it's an animal, all the organisms, they grow. Now here the two aspects, one is the quantitative and other is the qualitative. When you say quantitative, it is basically the length, breadth, the body mass, etc. Whereas the qualitative, that is as we see the root, seed, fruits or the plants as they get developed and differentiate into various parts of the plant body. So first part, the quantitative aspect, speaks for an increase in length, breadth, size, volume, body mass or dry weight and the number of cells. So these constitute the quantitative aspect. Further, growth as a quantitative change is a final end product of successive metabolism. So as the process begins from the growth of the seed into a sprout, further into seedling, and it grows into fully grown plant. So this continuous growth, or we can say successive metabolism taking place, is the final product of a plant growth. Qualitative aspect talks about the change in nature of the growth, where development is an ordered change or progress, while differentiation leads to higher and more complex state. So here we can see this qualitative being mainly the developmental aspect and the differentiation which leads to higher state of the plant body. Moving further, growth thus can be defined as permanent, irreversible increase in the bulk of an organism accompanied by the change of form. So as we know, growth is a permanent change, irreversible change which takes place in all the living organism, and can see the change in form of the organism. in multicellular, that is vascular plants, growth is indeterminate and occurs throughout the life indefinitely. So in case of multicellular plants, as we know, there's a small differentiation in a plant and animal. The plants, they grow indeterminate, continuously, indefinitely, whereas in case of animals, it's definite and determinate. Further, it is restricted to some specific region called meristems, which are the region where new cells are constantly and continuously produced. So in case of meristems, where new cells are formed and they continuously produce the new cells. Further, meristems are of three types based on the location, that is apical meristem, intercalary meristem and lateral meristem. So further in the slide, we can see all the three parts and what is the position of these three parts. Further, do you know roots and stem show indefinite or indeterminate growth as we just read. However, in organs like leaf, flower and fruit, growth is determinate. That is, they grow up to a certain genetically destined size. So as we know, the growth is indefinite or indeterminate in plants, but in case of leaf, flower and fruit, it is definite, and based on the type of plant or genetic composition of the plant, the size may be remain constant according to the plant or the species. Further, in unicellular plants, growth is uniform and determinate. Now this is what we spoke about is a multicellular, but in unicellular, growth is always determinate and uniform. Now, here I added a additional information. As you might have seen in all my videos, there's always additional information related to the topic, either images, animation, or both. So here I've added a beautiful and something unique information over here, that is Velonia ventricosa, a species of algae. Now all of you might have seen algae species. If I give you an example, Spirogyra, right? All of you might have heard, or Sargassum, bigger known species, right? But here, species Velonia ventricosa, one of the world's largest single cell organism. Now this algae which I posted the image is a single cell organism, but if you see the image, it looks like a balloon or a ball, very bigger in size, right? At full growth, it can be as large as a tennis ball. Scientists have been baffled by its structure for over a century. That means now algae, you might have seen how the plants, or as I gave you example, Spirogyra looks like, or a Sargassum looks like. But here it is a single cell which is very big, huge, like a tennis ball. So something which is fascinating as well as unique, and which actually even baffled the scientist, right? Moving further, do you know meristematic tissue? Again, we learn about three types as we just read earlier in the slide: apical meristem, intercalary meristem, and lateral meristem. So, apical meristem, in vascular plants, growth is restricted to the apices of the root and the shoot. So, growth is restricted for the root and the shoot region at the apex part. It is responsible for growth in length, height, and the differentiation or the cell types. It contributes the primary growth, that is the basic growth or the initial growth or the primary growth is contributed by the apical meristem, that is cell types or the length or the height of the plant. Second, intercalary meristem. Now, it is located at the node or at the base of the internode of the stem. It is primarily responsible for increasing length of the internodes and also formation of leaf primordia and lateral buds. Now, as the name intercalary meristem, it usually lies in between. As we read, node and the internode, which is usually the middle part in case of the stem, right? So it helps in increase the length of the internodes and also forming the leaf primordia or the lateral buds as well. Third, lateral meristem. It is located laterally along the axis of dicot and gymnosperms. It is located as a strip in vascular bundles of a stem of dicot. Now, the lateral meristem mainly found in the axis of dicot and gymnosperms, And in case of the dicots, mainly it is present as stream or a strip region in vascular bundles called as vascular cambium. It is responsible for increase in girth of the stem due to addition of secondary vascular tissue. So here I added the image, that is meristematic tissue, three types: apical meristem, intercalary meristem, lateral meristem. Here you can see the shoot apical meristem which is present at the apex region. Then you can see the root apical meristem which is at the bottom or the lower part, basal region. Here the intercalary meristem, the lateral meristem. The intercalary in between the nodes where it helps to elongate the internodes. Moving further, phases of growth. Cell in the meristem divide, enlarge and get differentiated. Corresponding to these three stages, there are three phases of growth. That is, first, phases of cell division or formation. Now, here again I added the animation image showing the root part. Now, from the basal region, the root cap. Further, you can see the different coloration which helps to understand the different region. The red showing the endodermis, blue showing the pericycle, the pink showing the phloem, and the green showing the xylem part. So you can see the phloem, which is the conducting tissue which conducts food, xylem water, and the pericycle and endodermis surrounding to the region. So this animation also helps us to understand how constantly and continuously the root keeps on growing below the ground. First point, cells of meristem are thin-walled, non-vacuolated, having prominent nucleus and granular cytoplasm. So the cell consists of non-vacuolated permanent nucleus with granular cytoplasm, that is cytoplasm in the form of grains which are granular, or we can say meristematic cell undergoes mitotic division to form two new cells. So here the meristematic cell undergoes division by mitosis. One cell remains meristematic and the other cell undergoes enlargement and differentiation. So by mitosis, when the cell divides into two, the one of the cell remains as a meristematic cell and the other cells undergoes the differentiation, forming the enlargement of the cell and the differentiation of the cell. Further, in this phase, rate of growth occurs at a slower pace, that is lag phase. Moving further, the second phase, that is phase of cell enlargement or elongation. The newly formed cell becomes vacuolated, osmotically active and turgid due to the absorption of water. Now, as we see the second type, it is cell enlargement, that is cell gets enlarged or elongated. So here the cells which is vacuolated, they are active osmotically, and as they absorb water, they become turgid. The turgidity results in enlargement of the cell lengthwise and breadthwise. So because of the turgidity, because it absorbs water, it becomes turgid and it enlarges both by length as well as breadth. In this phase, new wall materials and other materials are synthesized to cope up with the enlargement. So in this case, the new wall materials which are formed or the other materials which are present are actually synthesized or produced for the enlargement purpose. The growth rate in this phase occurs at an accelerated pace. Now, as you saw the first phase, which was actually slow, here it is at an accelerated phase and it is the exponential or the log phase. The third phase is the phase of cell maturation and differentiation. So here the cells, they mature or get differentiated. The enlarged cell now becomes specialized to perform specific function and attains maturity, both morphological and physiological. So here we can see the plant as they grow, the external appearance, which is the morphology, and the physiological appearance, that is inner part, both gets enlarged as well as specialized to perform the particular function of the cell. In this phase, the rate of growth slows down and comes to steady state. So here again it is a slow phase of growth, but it is steadily maintained, hence termed as stationary phase. So you can see here the image which I added, the figure 7.1 from the text, phases of growth in root, position of radical at the beginning A and at the end B. So you can see A, how the seed which get germinated, sprouted, the root formation takes place. That's why in the beginning, as I added a slide, simple slide showing the seed germination, radical, plumule, cotyledons. So you get an easy understanding as you learn further. Now, the B part, if you see, the root further elongates and plumule formation taking place, and you can see the different regions of the root. That is zone of mature cells, zone of cell differentiation, zone of elongation, and the zone of cell formation. Okay. Now, moving further, conditions for growth. So, what are the conditions required for growth to take place? Here, again, I added a simple image showing the plant. As the image shows, it's a sunflower. In presence of sunlight, it gets light energy. The oxygen which is given out by the plant, that is when photosynthesis takes place, so minerals and water absorbed through the root passes to the various parts of the plant. The carbon dioxide is taken in through the leaf. Sugar, that is a glucose, and oxygen is released during the process of photosynthesis. So now what are the conditions required for the growth? First point, the different environmental and physiological conditions necessary for the growth include water, as we know the most important aspect, supply of nutrients for the better development of various parts of the plant, maybe the fruit, maybe the flower, maybe the leaf, temperature, oxygen, carbon nitrogen ratio, gravitational force, light, and growth hormones, which all are important for the growth of the entire plant. The chief conditions are explained below. So we take each of the condition one by one. First, water, the most important essential for all the living organisms, is the essential component of protoplasm and maintains turgidity of the cell. It acts as aqueous medium for biochemical reaction. So as we know, the water is very important for all the living organism. In case of growth, they form the protoplasm of the cell and also the aqueous medium, that is water medium or liquid, where all the reactions takes place, or as we use the term biochemical reaction. Second, micro and macro element. When you say micro, the elements which are required in small proportion. Macro, the elements which are required in larger proportion, are nutrients required for proper growth of plant. Now, if any of these micro or macro are absent or not available subsequently or in a adequate proportion, then the growth of the plant may be affected. Further, third point, optimum temperature ranges between 25 to 35 degree for the growth of the plant. Oxygen is essential for respiration. As we know, during photosynthesis, oxygen is released, but for respiration purpose, oxygen is needed and the release of energy takes place during respiration. Then light, which is very much essential for germination of seed and photosynthesis, because if there is no light, no heat, no temperature, and the germination of seed may not take place, and thereby no photosynthesis. Then gravitational force decides the direction of growth of shoot and root. So as we know, because of the gravitational force, the root which moves inside the ground and shoot above the ground. Now, further, growth in plants can be measured in terms of. So, how the growth can be measured actually? First point, increase in number of cells produced. Example, single maize root apical meristem can give rise to more than 17,500 new cells per hour. You can see the increase in number of cells produced in 1 hour. the apical meristem of root, they can produce 17,500 new cells. See, isn't it amazing? 1 hour, new cells are produced at an amazing rate. Second, increase in surface area of the leaf. Example, growth of the dorsiventral leaf. As you know, the leaf are always dorsally, that's from the upper side, and the lower side they are flat. So the increase in surface area. Increase in length. Example, growth of the pollen tube. Increase in volume of fruit. Example, in watermelon flower, ovary after fertilization increases in size, volume by up to 3,50,000 times. Increase in girth of the shoot, that is the breadth, or we can say lateral meristem. Increase in dry weight of the organ. So these are some of the important points which helps to measure the growth in plants. Further, various methods for measurement of linear growth of stem and radicle are as follows. So there are more other methods which are useful or which can be used for the measurement of linear growth of stem and the root or the radicle. So what is that method? First is the direct method. It can easily be measured with the help of ordinary measuring scale. It is simple method. So using a simple measuring scale, we can measure it, right? It's a simple method. Then horizontal microscope. Now all the different measuring units, I had posted the image in the next slide so that you can understand what is this horizontal microscope or oxonometer or crescograph. So it will be easier for you to learn and understand with the images. Okay. Now, it is in case of horizontal microscope, it is used to measure growth in fields, that is agricultural fields or the plowing region. Oxonometer. This equipment is used for precise measurement of linear growth of shoot. There are two types of oxonometer, that is arc oxonometer and Pfeffer's oxonometer. So both the oxonometer images I posted in the further slide. Then next, crescograph. It records primary growth very accurately. Now as you know, graph which always records the movement, measurement, right? So here it is used to record the primary growth. It magnifies growth up to 10,000 times, giving information of growth per second. It was developed by Sir J.C. Bose. Now students, here you can see all the images which we just read earlier. One is the arc oxonometer. You can see how it is, the instrument looks like, the potted plant, and the arc oxonometer with the stand, the pointer, pulley, the arc to measure. Whereas the second type, Pfeffer's oxonometer. In this case again potted plant, you can see the stand, how is the position in comparison to the arc oxonometer and how the pulley is arranged. Okay. Then other than that, as we read, horizontal microscope. Now this one was discovered by or invented by Charles Chevaliers in the year 1834. Then triangular scale for the simple measurement. Here crescograph for measuring the growth by using the graph. Then I posted the image of Jagdish Chandra Bose, who invented the first crescograph in the year 20th century. So you can see how the invention was made in case of crescograph and in case of horizontal microscope by Charles Chevaliers. Now, growth rate and types of growth. Growth rate, it is the increased growth per unit time. It is also called efficiency index. Rate of growth can be measured by an increase in size and area of different plant organs like leaf, flower, and fruits. So the growth can be measured with the various parts of the plant organs, as we just read, leaf, flower, or fruits, right? And this rate of growth can be measured with the increase in size as well as the area. And this growth per unit time is termed as efficiency index. Further, the ratio of change in the cell number is given by dn over the time interval dt is called absolute growth rate, that is AGR. So students, AGR, absolute growth rate, it can be a one word term as well. Alternatively, it is measured and comparison of the total growth per unit time. So as we came across the terms AGR, absolute growth rate, is equal to dn by dt. What is dn? The ratio of change in the cell number, whereas dt, time interval. So AGR is equal to dn by dt. Further, the AGR, when divided by total number of cells present in the medium, gives relative growth ratio. Now students, another term, RGR. What is that? Relative growth rate. AGR, absolute growth rate. RGR, relative growth rate. Alternatively, RGR refers to the growth of a particular system per unit time, expressed on a common basis, or it is a ratio of growth in the given initial growth. So RGR is equal to AGR upon N. So we can create a ratio or understand the ratio of growth by RGR, that is relative growth rate, is equal to absolute growth rate upon N. So AGR and RGR are useful in describing the dynamics of cell growth in culture. So just a simple image showing the increase in the growth rate, how the growth rate increases of plant part, maybe leaf, flower or fruits. Now further, types of growth. There are two types of growth: arithmetic, geometric. So now just now as we learned the growth, the how the growth takes place, what all things can be used for the measurement. Now the growth can be put into two categories, that is arithmetic and geometric. Now both terms you are familiar, that is mathematical term, arithmetic, geometric. So you can have an understanding how the arithmetic growth takes place and how the geometric growth takes place. So first part, that is arithmetic growth. Here the ratio of growth is constant and an increase in the growth occurs in arithmetic progression, that is 2, 4, 6, 8 centimeters, etc. In this type of growth, the rate of growth is constant. So as I mentioned, you always have come across the term arithmetic, right, and geometric. So in arithmetic, it is in the multiples of 2, 4, 6, 8, or as we come across the table 2, the even number or whichever number it is, accordingly the number increases as given the example 2, 4, 6, 8, 10, 12, 14, so on, right? Then after mitosis, one of the daughter cell continues to divide and other cells takes part in the differentiation and maturation. Again, just earlier said, we learned mitosis takes place, so two daughter cells are formed in which one remains as a meristematic cell and the second one where the cell takes part in differentiation and the maturation. Example, elongation of root at a constant rate best explain arithmetic growth. So in case of arithmetic growth, as we saw the number 2, 4, 6, 8 means there's a constant growth. It is not directly from 2 it jumps to 5, 10 or 20, right? It is a sequential growth at a constant rate. Further, linear curve is obtained when growth rate is plotted against the time. So here when we observe this linear curve of growth rate against time, you can see how the changes takes place, which we will be seeing the graph in the further slide. Arithmetic growth is expressed mathematically by an equation. It is expressed as LT is equal to L0 plus RT. So to calculate this arithmetic growth, how the growth takes place, actually we have the mathematical formula which can be expressed as LT is equal to L0 by RT. Now what are these terms? LT indicate length at time t, L0 length at time 0, R growth rate, T time of the growth. So when graph of length L is plotted against the time t, a linear curve is obtained as indicated in the diagram. So we can see the diagram in the further slide. Now students, here you can see both the images which is there from the text. is graph 7.2 constant linear growth, which is given by W1 is equal to W0 ERT. W1 the final size, W0 initial size, R growth rate, T time of growth, and E base of natural logarithm. So you can see the graph plotted height of the plant L against time T. So you can see here the linear growth which takes place sequentially. Okay. Then here further we can see uh, what is the difference of, as just mentioned, the two types, arithmetic and geometric. So arithmetic, as you know, it is 2 4 6 8 goes constantly. Geometric, so you can see by the image itself how the multiplication takes place, how geometrically the growth of the plants takes place. So here you can see the zygote getting divided, the geometric phase of all these cells. Here the arithmetic cell divides, these cells divide. You can see the differentiation, the cells do not divide at this phase, initial phase. Cells capable of division, cells that have lost capacity to divide. So the diagrammatic representation, that is arithmetic and geometric. And C part, stages during development, that is embryonic development, showing geometric and arithmetic phases, as mentioned figure 7.3 growth rate from the text. Now coming to geometric growth, now as you saw the image, how what is the geometric growth actually and what was the arithmetic growth. Now the first point, cell divides mitotically into two. Here both the daughter cells continue to divide and redivide repeatedly. Such growth is called geometric growth. Now unlike arithmetic, as I told, a simple sequential method 2 4 6 8, here it is cell divides mitotically and the daughters, they continue to divide and redivide. Here the growth rate is slow initially, but later on there is a rapid growth at exponential rate. So the starting or initial growth of geometric is slightly slow, but once it starts, the growth is very rapid at an exponential rate, means the growth is very faster. So geometric growth can be expressed mathematically by an equation, W1 is equal to W0 ERT. Now what are these terminologies? W1 the final size, W0 initial size, R growth rate, T time of growth, and E base of natural logarithm. So again here I had posted the image from the textbook, that is figure 7.4 growth rate comparison. Now you take any plant leaf, smaller one or bigger one, the growth rate always remains the similar in case of arithmetic or geometry. So here the growth rate, if you see the earlier image of the leaf, 5 centimeter square, and it is further growth is 10 centimeter square, whereas in B 50 centimeter square, and here it is 55 centimeter square. We can also observe quantitative comparison between the growth of living system in two ways. So even the quantitative comparison can be done in two ways. Measurement and comparison of total growth per unit time is called absolute growth, AGR. Just in the earlier slide we came across the term AGR and RGR. So what is AGR? Measurement and comparison of total growth per unit time. Whereas growth of given system per unit time expressed on a common basis per unit initial parameter is called the relative growth rate, that is RGR. Further, in the above example, two leaves A and B are of different sizes but show both absolute increase in area at a given time. Both leaves grow and increase their area by 5 centimeters square to produce A and B leaves. So as I mentioned, A and B, two leaves, one is smaller and big, but the growth rate is similar in both. So here both increases by 5 centimeters square, that is from 5 centimeter the natural length grows to 10 centimeter, and here the leaf from 50 to 55. So there is a difference in 5 centimeter in both. Further, growth curve, it is a graphic representation of the total growth against time. So always a graphical representation is growth against time. there are three types of curves, that is linear, exponential and sigmoid. Arithmetic growth curve is linear while geometric curve is exponential. So as per the growth curve, the three types are the linear, as the name suggests, in a linear manner, exponential at a high rate, and then the sigmoid, that is, you can say S-shaped curve. Arithmetic curve as is usually the linear one, whereas the geometric curve is for the exponential one. As arithmetic goes sequentially, as we learned 2 4 6, geometric in a high exponential manner. Corresponding to three distinct phases of the growth rate differs. In lag phase, growth rate is slow, which again you saw in the earlier slide. Exponential log phase, that is growth phase, is faster and reaches to its maximum. In stationary phase, growth rate gradually slows down when a graph of rate growth against time is plotted for three phases of growth. A sigmoid growth curve is obtained. The total time required for all phases to occur is called grand period of growth. So as you can see here, the three phases, log phase, lag phase and the stationary phase, and this time take total time required for all the phases is grand period of growth. So come to the image exponential growth. Here you can see the population size and the time how the growth phase takes place. Here in the second image, you can see size, weight of the organ and the time. So here the lag phase, how it starts slowly, gradually, and then it grows at an exponential rate, faster rate, and after a certain time period it becomes stationary. Moving further, differentiation, de-differentiation and redifferentiation. Students, until now we might have come across the term differentiate or differentiation, right? To differentiate part of the plants or part of the flower. But now here we also learn further about de-differentiation and what is redifferentiation. So first part, differentiation. It is maturation of cells derived from apical meristem of root and shoot. So here maturation takes place in the apex part of the plant of shoot and root part. Permanent change in structure and function of cells leading to maturation is called differentiation. So as we can see here, the term permanent change. So here change occurs permanently in case of the structure and function of the cells, which further leads to the maturation. During cell differentiation, cell undergoes few to major anatomical and physiological changes. Example, parenchyma in hydrophytes develops large cystogenous interspaces for mechanical support, buoyancy and aeration. So here we can see example, parenchyma in hydrophytes. The term hydro means water, phytes plants. So water plants. So parenchyma of in water plants, how we can see the differentiation which helps them for mechanical support, for buoyancy, that is in floating in water, and for aeration, which you can say help in taking up oxygen. The maturation is at the cost of capacity to divide and redivide. So it helps not only to mature but also further to divide and redivide. So here you can see TS of parenchyma, that is transverse section, the nucleus of the parenchyma cell, the intercellular space, vacuoles and cytoplasm. Here we can see the image which I added, the examples given in the text, that is free floating hydrophyte. So here you can see the parenchyma and hydrophyte. So I have posted the image of Echinoria pistia. You can see both plants floating in water. The roots also floating in water with the root pocket, fibrous root, the stolon and the floating leaves. Even in case of pistia, we can see the root pockets, fibrous root, offset. This term we are familiar, which we learned in the topic morphology of flowering plant. Inflated petiole lamina, and C and D, that is Nymphia Marsilia, root floating hydrophyte. So you can see the root which is embedded in the soil, the flower bud, petiole which is very long, which helps the lamina, that is a leaf, to float in the surface, and the flower. Here also you can see in case of Marsilia, the leaflets, petiole, the young leaf, the rhizome, root, mud where the rhizome and the root are embedded. Moving further to the second part, de-differentiation. Now, as we learn differentiation, which all of you have come across, now de-differentiation. The living differentiated cell which has lost the capacity to divide may regain the same as per the need and divide. Now, we have seen that differentiation takes place, that is a cell, they divide and they get matured. But there are certain cells which after getting differentiated, they again may divide as per the need of the plant. So in that case, it is de-differentiation. Thus, permanent cell undergoes de-differentiation and becomes meristematic cell. So as we know, as we have learned in the chapter morphology in FIJC, also as we are learning here, the meristematic cells which usually divide and redivide. But once they divide and redivide to form any organ, they become permanent. But here, this permanent cells further undergoes de-differentiation and they become meristematic cell. Example, in interfascicular cambium and core cambium are formed from parenchyma cells between vascular bundle and innermost layer of cortex respectively. So to understand this concept, again I have posted an image over here. First you can see the plant, shoot tip, shoot apical meristem and young leaves, lateral meristem, vascular cambium, core cambium, axillary bud meristem and the root apical meristem. Here we can see the inner section, how it appears. So that is what we learned, the de-differentiation intervascular cambium and the core cambium. Here highlighted with the red color. So here vascular cambium, secondary xylem, primary xylem, pith. Here you can see the secondary phloem, primary phloem, cortex, core cambium and the periderm. Now again in the FIJC usually do the practical making the section of sunflower stem or cucurbita stem, right? Primary growth in the stems, that is which you can see from the young leaves, epidermis, cortex, primary phloem, secondary xylem, pith, which here we can see the not only the primary but secondary xylem and phloem. Moving further, the third type, redifferentiation. From differentiation to de-differentiate to redifferentiation. The cells produced by de-differentiation once again lose the capacity to divide and mature to perform special function. This is called redifferentiation. Example, secondary xylem and secondary phloem are formed from de-differentiated cambium present in the vascular bundle. Now students, I think now you might understand this meaning of de-differentiation, redifferentiation. Just to recap it, de-differentiation, the cells which are already matured, they lose the capacity to again differentiate, and they as a differentiate, that is they become de-differentiate. Now those which de-differentiate, these cells, they divide and mature to perform a specific function, that is they re-differentiate. So here the example of it is secondary xylem and secondary phloem. Just as I mentioned in the previous slide, the image which I added, where I also showed the secondary xylem and phloem. So here also again I added another image for your better understanding. So you can see this image, primary phloem, primary xylem, which is already present. Intervascular cambium, vascular cambium. This is what is de-differentiation which we saw. Vascular cambium, cortex and epidermis. Now if you see in this the second image, epidermis, cortex, primary phloem, secondary phloem, vascular cambium, here primary xylem, secondary xylem and the pith. So here the secondary phloem and the secondary xylem, they are the redifferentiated cells. Clear students? So that is the last part for the first part of the chapter. I divided this part into three parts. The first part we have completed, and based on that the assignment question, short question for 1 marks, 2 marks short note and 3 to 4 marks long answer. Thank you students.